Welcome to the news report, the race for the structure of DNA, theorists, experimentalists, and the future. Today, the host will be
Sarah.
Iris.
Fabia.
and Eric. It's 1950s, and the time was prime for a breakthrough, the structure of DNA. It begins with Watson and Crick, guys who bonded over a shared inspiration for DNA. The two collaborated at Cambridge University, but they soon learned that they have competition. Collaborators Wilkin and Franklin and Linus Pauling, who all wanted to be the first to discover the structure of DNA. The race starts in 1951 with all teams independently working. Watson and Crick's method of a theoretical approach had the advantage of quickly making hypotheses and theories based on existing data. They soon made their first model, but it was wrong and criticized by Franklin. The model was inside out and had wrong chemical bonds. This is not shown in the image. After this, the head of the lab banned them from working on DNA, telling them not to fish in other people's ponds. This highlights the disadvantage of their method, their dependency on other scientists' data and the theories that are often wrong. They had to go through the long, hard, and repetitive process of collecting data from various sources and then making theories that are most likely wrong.
On the other hand, Franklin and Wilkins use X-ray diffraction. The advantage is that it provides precise atomic levels details of DNA structure. However, the disadvantage is that the process was long, hard, and repetitive, taking tens of photos to get the right one, and exposure to radiation likely contributed to her death later. Yet, Franklin's intuition and inspiration based on these painstaking photos led to her suspect the double helical structure of DNA long before it was confirmed. Wilkins and Franklin also had a strained relationship. Franklin was independent, Wilkins was assertive. In January of 1953, Linus Pauling revealed his triple helix. Watson and Crick realized they had to hurry. So Crick goes to someone to confirm Chargaff's base pairing ratios, and Watson goes to Franklin to bet for her photos. She says, "You all think this is a game? This is an example of Watson and Crick being driven by their inspiration to be the first. Franklin was driven by her inspiration to be accurate, which clashed."
It wasn't until Wilkin leaked Photo 51 to Watson, old notes about DNA strands being anti-parallel, and modeling bases with paper cutoffs that they finished the structure. These moments are all examples of Watson and Crick's intuition turning evidence into results. On April 25, 1953, Watson and Crick published their DNA double helix with all the correct details. Their work can be simpler visualized with this model. Their win was controversial. Many believe that Watson and Crick couldn't have done it without Franklin's Photo 51. Years later, Watson and Crick were awarded the Nobel Peace Prize. Franklin was served one, but she died before it was given. The discovery of DNA structure is important. It contributed to society at the time by opening up new fields for molecular biology and genomics. More specifically, scientists later discovered DNA replication, where both strands of the original DNA are split and two complementary strands of DNA are made, one for each, ending with two identical DNA strands. RNA transcription, where DNA is copied into a complementary mRNA strand. Translation, where tRNA reads the mRNA and brings the corresponding amino acids. And then the next tRNA would bind to the remaining mRNA to form a chain of amino acids to form a polypeptide chain, which will fold into proteins.
RNA differs from DNA by using ribosome sugar and replacing thymine with uracil. Similarities that are transcriptions and translation rely on a template sequence, and DNA and RNA are both chains of nucleotides. These processes and molecules connect back to Watson, Crick, Franklin, Wilkins, Pauling, and Chargaff. Chargaff's base pairing ratios were essential for discovering DNA replication, and Franklin's X-ray revealed the double helical structure of DNA that makes transcription and translation possible. Wilkins' leaking of data and Pauling's competition helped drive Watson and Crick forward, hastening their publishing of the structure of DNA, a breakthrough that helps scientists understand the works of RNA and the making of proteins. The paper models that are from the DNA RNA protein lab, and the purpose is to help construct DNA, shows replication, and teach the names of molecules in DNA and RNA. It demonstrates transcription, the matching of tRNA and mRNA bases, how tRNA bring amino acid to ribosomes to build protein translation and mutations. The lab is done with the materials of paper models and scissors. Nowadays, modern society uses the discovery of DNA, RNA, protein, replication, transcription, and translation to better our understanding of genetics. These fields include possible advancements in the future, such as genetic editing like CRISPR, stem cell therapy, and RNA-based vaccine. And that wraps up today's news, the history of discovery of DNA. Goodbye and thank you for tuning in.